Hello everyone and welcome back to the Advanced Higher Biology course. Today we are going to look at key area 2BI, which is intracellular membranes. Let's get started. The three learning intentions today are to understand the difference between prokaryotes and eukaryotes, to understand the importance of internal membranes as they increase the total area of the membrane, and finally to understand the structure and function of different internal membrane structures within the eukaryotic cell. From higher biology, you will be aware of the three domains of life. Remember, they spell out BAE: bacteria, archaea, and eukaryota. We are going to look at the membranes within the cells of these domains of life. Only the single-celled organisms of the domains of bacteria and archaea are classified as something called prokaryotes. The pro means before and the kary means nucleus, so it's before the nucleus. Prokaryotes are simple, single-celled organisms that lack a nucleus and they lack membrane-bound organelles. Prokaryotes are not split up into different organelles, so they don't have things like mitochondria. They consist of one open space that contains cytoplasm and things like genetic material. Eukaryotic cells, however, have a system of internal membranes that increases the total area of the membrane. Unlike prokaryotes, eukaryotes have a membrane-bound nucleus and a number of membrane-bound organelles. Cells of prokaryotes are much smaller than that of eukaryotes. You can see that on this logarithmic scale. So bacteria on the scale, which are prokaryotes, are roughly around 1 micrometre in size, whereas animal and plant cells, which are eukaryotes, are between 10 and 100 micrometres in size. As you can see from this table, typically the diameter of a prokaryotic cell is between 1 and 5 micrometres, while eukaryotic cells are larger and they're between 10 and 100 micrometres in size. Okay, so now we've looked at the difference between prokaryotes and eukaryotes. We are now going to look at how we understand the importance of internal membranes as they increase the total area of the membrane. So this is probably our trickiest learning intention today. Eukaryotic cells have many metabolic processes occurring within them, such as respiration, protein synthesis, and photosynthesis, to name a few. The cell does not simply complete these processes in isolation. They require raw materials to be passed through the plasma membrane into the cell to start these metabolic processes, and waste materials to be removed via the plasma membrane after a metabolic process has finished. Let's have a look at the difference between a smaller prokaryotic cell and a larger eukaryotic cell. When a cell is smaller, like in this example, it has a smaller volume. But when you compare the surface area, in cells the surface area is just the area the plasma membrane takes up, to the volume of the cell, it actually has a larger surface area to volume ratio. So a small cell has a large surface area to volume ratio. This is good for the cell as there is enough surface area in the plasma membrane alone to provide raw materials and release waste products for the volume of the cell it has. When a cell is larger, like in eukaryotes, it has a larger volume, which makes sense. The surface area also gets bigger, but importantly, not by that much. This means that when you compare the surface area, which again is the area the plasma membrane is taking up, to the volume, it actually has a smaller surface area to volume ratio. So if you've got a large cell, it's got a small surface area to volume ratio, so it's the opposite. This poses a problem to larger cells, as this means that there may not be enough surface area to provide raw materials and release waste products for the volume of the cell, and thus the amount of metabolic processes that are going on within that large cell. Okay, so I've talked a lot about surface area to volume ratio, and I'm going to explain to you what that means. Now, you don't really need to know in depth what that means, as long as you know a large cell has a smaller surface area to volume ratio, then that's fine. But I'm going to kind of talk you through the calculation just to give you a feel for it. So, in this example, we have got a cube which is just representing a cell. The surface area of this cell is 6, whereas the volume is 1. The surface area to volume ratio would just be 6 divided by 1, which is 6. Smaller cells generally have a larger surface area to volume ratio than larger cells. So this small cell has a surface area to volume ratio of 6. Now if we compare that to this larger cell, so again the cube is our larger cell, the surface area in this large cell is 96, whereas the volume is 64. So to get the surface area to volume ratio, you need to divide 96 by 64, and that gives you 1.5. So our surface area to volume ratio is 1.5. Now when you compare that to the previous one of 6, it shows that larger cells have a smaller surface area to volume ratio. Okay, so this is just a table to summarise. So a prokaryote cell type has a larger surface area to volume ratio. This means it has enough surface area in its plasma membrane alone to cope with all the metabolic processes that occur within the cell. A eukaryotic cell type has a smaller surface area to volume ratio. This means it can't cope with all the metabolic processes going on within the cell. They need a solution to increase the total area of membrane for vital metabolic processes. And there is a solution. Large eukaryotic cells have a system of internal membranes which increase the total area of membranes within cells for your metabolic processes. Okay, so the final learning intention is to understand the structure and function of different internal membrane structures within the eukaryotic cell. Large eukaryotic cells have 4 internal membrane structures that increase the total surface area of the cell. The first is known as the Endoplasmic Reticulum, or ER for short, then the Golgi Apparatus, then the lysosomes, and finally the vesicles. The Endoplasmic Reticulum, or ER for short, is attached to the nuclear membrane. It is a network of membrane tubules that can either be rough, which is known as the rough endoplasmic reticulum, or smooth, known as the smooth endoplasmic reticulum. And you can kind of see that in the diagram. So the blue in the right hand side diagram is the rough endoplasmic reticulum because it's got all these dots on it, which are, you'll find out, are ribosomes. And the smooth endoplasmic reticulum is the orange, and you can see that it doesn't have many ribosomes on it. The Golgi apparatus is a series of flattened membrane discs, which we will later find out have a role in the post-translational modification of proteins. The Golgi apparatus is usually found between the endoplasmic reticulum and the plasma membrane. Vesicles transport materials such as proteins between membranous compartments. The diagram on the right hand side shows this clearly. we see that the vesicle has firstly taken the material from the endoplasmic reticulum and transported it to another membrane compartment known as the Golgi Apparatus. Another vesicle has taken materials from the Golgi Apparatus and transported them to the plasma membrane to be released from the cell. Vesicles, as you can see from the left-hand side image, have a bilayer of phospholipids, which shows you that they are intracellular membrane structures. They're made of membrane. Lysosomes, again, are membrane bound. They also contain an enzyme called hydrolase or hydrolases. They can digest many materials such as proteins, lipids, nucleic acids, and carbohydrates. Okay, so our 3 learning intentions today were to understand the difference between prokaryotes and eukaryotes, to understand the importance of internal membranes as they increase the total area of the membrane, and to understand the structure and function of different internal membrane structures within the eukaryotic cell. Now that we've went through the PowerPoint, these are the revision tools we're advising you to use. So the summary notes can be found in the file section of Teams or in our assignment. If you look at page 38, this gives you all the information you need this week about intracellular membranes. I've also attached scans of a revision book for you to read. Finally, if you type in Study Stack and in the search bar type St Ninian's HS, this will come up with loads of flashcards. Click on the Advanced Higher Synthesis and Transport of Proteins. I know that doesn't make sense because it's intracellular membranes, but that's where all the questions are, to get the flashcards for intracellular membranes. Okay, so because we've got a holiday this week, I'm going to extend the assessment materials into next week. So next week you're going to get revision questions and you're going to get an intracellular membrane quiz. Please let us know if you're having any trouble accessing documents on Teams. I've also put a survey monkey up, so if you want to answer the questions on there to help in your home learning experience, then that would be really helpful, because we'll have a look at them and see if we can improve anything that we're doing in the biology department just to help you out over the next few weeks. And yeah, thank you for listening.